Hi, and welcome to my OCR A-Level Biology Revision session with me, Christine. So today's lesson I want to look at the convoluted tubule, which is part of your kidneys in the excretion module. So let's just remind ourselves about the kidney nephron then. So we have our glomerulus and our Bowman's capsule where ultrafiltration occurs. We then move through to the proximal convoluted tubule where selective reabsorption occurs, then down through the loop of Henle, which is necessary for water reabsorption, into the medulla, and then we get up to the distal convoluted tubule, which is where we're going to focus today's lesson. So let's just look at the distal convoluted tubule then. So one of the key things to note is that the distal convoluted tubule and the proximal convoluted tubule are different in their shape. So we can tell the difference by looking at the size of the lumen and also looking at whether or not there is a brush border which increases the surface area. So the distal convoluted tubule does not have the increased surface area of a brush border, and it has a slightly larger lumen. But it's got a very specific and similar role to play like the proximal convoluted tubule. So the distal convoluted tubule then is the site of selective reabsorption of mineral ions, such as sodium, chloride ions, calcium ions. Now, what we need to remind ourselves is that to be able to move these substances, what we have to first do is what's known as active transport. We need to create a concentration gradient, and we do that through the sodium-potassium pump. So the key thing to remember is that we will always use the sodium-potassium pump to move 3 sodium out and 2 potassium in, and that therefore is going to create a concentration gradient. You're going to have more sodium outside the cell and more potassium inside. Now let's just remind ourselves in the fact that these are membranes that go around, and therefore they contain proteins that are very specific. They are selectively permeable biological membranes, and therefore because they're selectively permeable, they will only allow ions to move across the phospholipid bilayer if there is a specific channel to allow this polar molecule to move. So potassium can actually diffuse down its concentration gradient out of the cell because there are potassium channels within the cell membrane. Well, the sodium has obviously moved out of the cell, and because the sodium and the potassium have got charges to it, what you tend to find is that there is a potential difference between the inside of a cell and the outside of a cell. Now, if we have any ions at a higher concentration on one side and a channel that allows those ions to diffuse, they will diffuse from an area of high concentration to an area of low concentration. But that will only allow it to occur on the side where the channel is present. So if we have, for example, the need for our calcium to be moved into the blood, if we selectively need to reabsorb that, what we do is we do that through the use of a co-transport protein. Now, the co-transport protein is going to move both the sodium and the calcium across the membrane. And it does that because the flow of ions down an electrical chemical gradient will drive the movement of the other ion. Now, be careful, active transport needed it to have the ATP molecule being hydrolyzed down to ADP, releasing the energy for the pumping. The co-transportation is to do with this electrical chemical gradient. The fact that these sodium ions are going to diffuse down their electrical chemical gradient into the cell, and that's therefore going to provide the motive force which allows for the calcium to be pumped out of the cell. Now, you don't need to know this in specific details, but they have in the past thrown a question in there about comparison of the distal convoluted tubule with the proximal convoluted tubule. So they do expect you to know the proximal convoluted tubule, the sodium-potassium pump, the active transport, and the co-transportation. They may give you a question with some information about what's happening in the distal convoluted tubule, and if they do, what they're expecting you to be able to do is to actually analyze where there's similarities and where there's differences. Now, it's not just about the movement of those ions across the membrane that we need to consider. We also have to consider the role that our body takes in controlling what might be our water potential, which is linked to the concentration of those ions in the blood. So if we have a blood which has too much water or too little salts, if our water potential of our blood is raised, then that's going to cause pressure inside our blood arterioles. Now, it's important for us to note that the excretion module links very much to the animal response module and also links very much to the hormonal system. So they'll never leave it as a standalone topic area. You need to find your links. So if we have too much water or too little salts, if our blood pressure is rising because our water potential is rising, this is going to be detected by receptors in our carotid arteries. Well, they're then going to have to signal to the adrenal gland, which is found at the top of the kidneys. What will then happen is the cortex, the adrenal cortex, is going to secrete the hormone aldosterone. Now, aldosterone is going to be transported in the bloodstream to the kidneys, and what it's going to do is it is going to signal for the kidneys at the distal convoluted tubule for those sodium ions to be reabsorbed. Now, aldosterone is a steroid hormone, and that means that it can actually bypass the membrane and get into the nucleus, causing transcription to occur to produce more sodium channels. Now, you'll learn about that in Module 6 in more detail, but please note that it's never one topic area on its own. If we have more sodium ions being reabsorbed into the distal convoluted tubule, what we're going to do is have our sodium concentration rise. We get back to that correct water potential. So this is an important role to ensure that our water potential is maintained at the right level. If we go the opposite way, if we've got too much salt, too little water, we lower our water potential. Our blood pressure is going to then fall. That's going to be detected. The adrenal gland is going to be signaled to release less aldosterone. If less aldosterone is being secreted, our sodium concentration in the blood tends to fall because we will excrete it, and therefore we maintain that correct water potential. I hope you've liked this video, and if you have liked this video, then please do click on the like button and subscribe to my channel. Also, if you haven't done so already, do check out my www.aiqchat.com revision platform.